Okay. Thank you all for staying, by the way, and you're a hardy group, so I'll try and keep it brief so you can go out. I have nothing really to disclose. So just, I got interested in this subject. I'm a professor of pediatrics and also allergy and immunology, and I got involved in this during the AIDS epidemic. We were thinking about vaccines for AIDS, obviously, and also chronic infections, and since obviously we don't have an AIDS vaccine, and we certainly are not very far along with herpes virus vaccines, I think it's still a topic, and maybe more so if we start to really cement some of these conclusions about a role for the EBV and other related things. The next question is, well, then what about antiviral therapy? What about vaccines, maybe T-cell vaccines? So supposedly the first reference to herpes viruses is in Shakespeare, which I think is interesting. The last line, it says, which oft the angry Mab with blisters plagues, because their breaths with sweetmeats tainted are. So Mab is a fairy queen who appears in Romeo and Juliet, and obviously there's always been a connection with sexually transmitted diseases, poor choices of sexual partners and practices, and I think this has a huge stigma, and I think, as I'll mention, you have to be ready for this whenever you discuss this with patients. But also there's a reference here about courtiers and lovers' brains, which we know that at least the simplex viruses do cause encephalitis, may play a role in triggering autoimmune brain syndrome. So it's just interesting that Shakespeare seems to have gotten it right long before us. But there are a lot of common fallacies. As I mentioned, when you talk to any patients about this, you have to reinforce that most herpes viruses, certainly the ones involved in common autoimmune disease like Epstein-Barr virus, I mean, it's called the kissing disease, but certainly roseola caused by herpes 6 and chickenpox, should not really be any stigma attached to these diseases. Another important fallacy is that viral reactivation, you need to have a positive IgM, and I think that many people who are not interested in this particularly as a specialty, this is just not true, and particularly with the gamma and beta herpes viruses, such as Epstein-Barr virus and herpes 6, what you're looking for is viral reactivation, which may involve expression of vesicles and genes that have nothing to do with generating IgM, and these viruses live in the immune system anyway. You don't have to confirm viral reactivation by PCR. If PCR is only useful, for example, in severe drug allergies, you may be able to detect viral reactivation, graft versus host, but very often these are just short bursts of viral replication and expression of genes. And the fact that many syndromes don't respond to antiviral agents is not a proof really of anything, because our antiviral drugs mainly target viral replication rather than viral gene expression. And in fact, there is a literature that I've reviewed about antiviral drugs in autoimmune diseases like MS, which I think is very intriguing, and I think it's sort of been ignored because many of these drugs like acyclovir are not very exciting and are off patent, but there is some signals that you may not treat the symptoms of the disease, but there may be some effects on viral progression. So just briefly, there are 8 known human herpes viruses. Alpha is in the neurons, gamma virus, this is what they were discovered, Epstein-Barr virus is in B lymphocytes, and then the beta herpes viruses, CMV, are all, herpes 6 and 7, are resident in stem cells, which I think is really fascinating, because they just kind of sit there until that particular stem cell activates, and then they replicate at a high level. So this is from a paper that I published in 2011 regarding the fascinating effect that EBV has on endogenous retroviruses. And endogenous retroviruses is a whole separate area that I'm not going to try and cover today, but in fact, both Epstein-Barr virus and herpes 6 activate endogenous human retroviruses. So even though they're exogenous in infections, they're activating viruses in us. So it raises a lot of questions about what's our self and what's not. So this has all been reviewed, and I'm not going to spend a lot of time on it except to say that the interesting thing about this is that the way that Epstein-Barr virus activates these latent herpes viruses, latent human endogenous retroviruses, is that they actually encode a homologue of CD40. And it turns out that actually the endogenous retroviruses are activated by our CD40, and the virus just has a copy of our gene. And so that's why I came up with this idea of gene sharing, which is a little different than molecular mimicry. Just to clarify that, molecular mimicry means that there are just certain epitopes that are shared between unrelated proteins, whereas in this paradigm, the viruses have whole genes from us, and really large numbers of them. So I'm not going to talk about all of them, but I'm going to spend a little bit of time on BCRF1, which I think is very fascinating because it's a homologue of interleukin 10. And if you've been at this meeting, you know that interleukin 10 has been coming up a lot, and it's going to come up more because it seems to be the link between the gut microbiome and immunosuppression in systemic. And then if we have a little time, I'll talk about the zebra protein, which is BZLF1, which is a DNA binding protein, which may have a role in anti-nuclear antibodies. I'm not going to have time to talk about EBNA and BALF2, but anyone who's interested, we do have a homologue of the RAG recombination activating gene, and EBNA is a very fascinating gene with many chromatin DNA binding. So this is just a review, which I think this talk is going to be in the journal article that comes out. But so the review is cited, but it basically IL-10 is encoded by many viruses, and it basically is very similar. This is just the I'm not even going to try and use it. Maybe I could use the pointer. Whoops. Nope, shouldn't have done that. Okay, so basically these viral IL-10s look just like IR IL-10s, and both cytomegalovirus and Epstein-Barr virus have a copy. Human herpes 6 has a copy of interleukin 6. And these seem to have resulted from numerous separate events, although the CMV and the Epstein-Barr virus IL-10 seem to have come from a similar event based on the gene structure. Many of the fish herpes viruses have IL-10s that seem to come from completely different phylogenies, and you can just very, very well written review. So this is important because IL-10 is a master regulator. It's the anti-interferon, if you want to think about it that way. It actually binds to the interferon receptors, but in some way turns them off, and that's why it has suppressor and T helper 2-like properties. And it's captured by many chronic viruses, and many other viruses have ways to activate IL-10. And why they do that, we could speculate about. Obviously, the virus wants to suppress the immune system that's trying to kill it. So this is just a figure from the, this is going to be in the journal, I hope. But just thinking about this, first of all, the viral IL-10 is similar to the human IL-10, but it's not identical, and it could be blocking the human IL-10. And one of the paradoxes of lupus, if we use that as a paradigm, is that you have very high levels of endogenous IL-10, which seems paradoxical, so something must be blocking it. So one possibility is just that the viral IL-10, which we're all making in this room, and goes up and down depending on our stress level and so on, is just blocking the host IL-10. The second possibility is that we all think that we're very smart because now we have anti-cytokine antibodies, but it's certainly not implausible that we make an immune response against the viral IL-10. Well, then what happens? So you get blocking antibodies. So this is, it should be also not a surprise at this meeting. And in fact, so 25% of healthy blood donors have antibodies against the cytomegalovirus IL-10. Nobody has ever characterized them. And one reason I'm giving this talk is anyone that's interested in this, I'm trying to make peptide arrays to look at these viral IL-10s and so on, and receptor antibodies. And obviously that could explain the upstream of everything, why EBV always comes up, and CMV to a lesser extent. But, and again, we know that herpes 6 has interleukin 6, so if you start getting anti-cytokine antibodies and so on, you could see that this could kind of spiral out of control. So it's a master regulatory thing. And the third thing, just to quickly, so the model that I would propose is that you have some upstream events, like the host, us, loses IL-10 because of the microbiome and so on. The virus replicates more. You start to get antibodies against the viral IL-10, blocking more IL-10, and then you could see how things could go wrong. So the last thing that I want to talk about quickly in terms of another fascinating idea about shared genes is the zebra protein, which I've published. This is all published, but it's a multifunctional protein, and the carboxyl terminus is a ankyrin-like region, and the domain 1 and domain 2 are captured from a human phospho transcription factor, which is what activates AP1 genes and interleukin-2 stimulation and so on. So this protein has been partially crystallized, and on the right side is the tweezer-like phospho that binds to DNA, and on the left, this blue region is what I call ZANK. So very quickly, this is the first known case of a correlation between a known HLA allele and a particular viral epitope. The HLA-B allele binds to this particular epitope, the shorter form of it, and this emission of 4 amino acids, EECD, basically makes it bind to another HLA, and these are directing the T cell response. But this actually shows, we know that HLA always comes up first. So this is actually a viral protein that is binding to DNA. It's a mimic of a host phospho transcription factor, and it's binding in a different way in people that get lupus versus people that don't. And so we know this is, like, I think it's very important. The blue region, I'll just very quickly mention, it's an ankyrin-like region. Ankyrin is involved in P53, IKB, BCL, lymphoma genes, and who knows what antibodies to that could do, but we know that certainly anti-nuclear antibodies are a hallmark of lupus. And so the gene sharing summary idea is a new paradigm that I'm kind of proposing, but that, you know, EBV is not, as I say, it's in most of us in this audience, and it's evolved over millions of years in a certain environment. So now we've changed that environment with the processed foods, with everything's different in our diet, and obviously the viruses respond differently to that. And if we start to lose IL-10, and these viruses all have IL-10, then we could certainly start a cycle where the whole thing kind of goes out of control. And I think it just emphasizes that in the future, if we really want to have drugs that treat these diseases rather than just modify our response, we're going to have better vaccines and maybe antiviral drugs that actually target some of these problems. So I'll just stop there.